So Campy just sounds too friendly. It sounds like a friendly little mascot
Yes.
rather than a deadly pathogen. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klinowski, your host for today's episode. When we hear the word carbohydrate, the first thought we might have is about our diet. However, carbohydrate molecules have many roles beyond our diet and are utilized by virtually all forms of life on the planet, including pathogenic bacteria.
Our guest today is Dr. Christine Szymanski, professor in the Department of Microbiology at UGA and a member of the CCRC, the Complex Carbohydrate Research Center. Dr. Szymanski has worked extensively on Campylobacter jejuni, a bacterial pathogen, and is an expert on the glycobiology of microbial pathogens. Christine, welcome to the podcast.
Thank you. I'm excited to be here.
Well, we're excited to have you. We thought we might kick off the podcast by first asking you to tell us about Campylobacter. Can you just tell us about how one is infected with this bacteria and what is the treatment for it?
Absolutely. So Campylobacter is known as a self-limiting diarrheal disease organism. And so typically in healthy individuals, we are able to clear Campylobacter ourselves. But in some cases, antibiotics are required, and also in some cases, some unfortunate individuals develop paralysis after Campylobacter infection. And so I'm sure you'll be asking me more questions about that. The source of Campylobacter in many places is poultry, contaminated poultry. And the reason for that is because Campylobacter is actually a natural gut commensal in birds, including poultry. And so undercooking your poultry meat will lead to Campylobacter infection and then subsequently to diarrhea.
It's really interesting that it's a normal commensal in one species and a pathogen in another.
Correct. And it's estimated that as few as 500 bacteria can cause disease in humans, and yet birds as well as pets, so cats and dogs, are also carriers of Campylobacter, and you can get Campylobacter infection from having your pets lick your face if they're also colonized with Campylobacter. So that's something to keep in mind.
So you mentioned that it is a natural pathogen, or not pathogen, wouldn't be a pathogen. It would be a natural part of the microbiome in chickens. So is that an approach that you're taking to basically control Campylobacter infection?
Right. So this is where the carbohydrate part comes into it, is that bacteria, unlike humans, have a lot of unique sugars that they decorate their surfaces with. And so it's actually advantageous to us to design vaccines that target these specific sugars. And so we're making what's known as glycoconjugate vaccines. And we're targeting two populations. One is chickens, to see if we could reduce the number of Campylobacter in poultry before they are seen by the consumer. But we're also looking at whether we can develop vaccines for humans, and particularly infants, because what's been shown recently in studies that have been funded by the Bill and Melinda Gates Foundation is that in low and middle income countries, infants are at very high risk of Campylobacter jejuni infection. In fact, one study showed that up to 85% of infants are infected with Campylobacter by the age of 1. And that has not only serious immediate effects, but long-term effects in causing growth stunting. And so there's a demand for vaccination of infants, but also for travelers. So if you go to the Center for Disease Control website, the CDC, it's estimated that Campylobacter infection occurs in about 1 in 200 individuals each year. So that's a very high rate of infection. And Campylobacter, because of those numbers, is predicted to be the leading cause of bacterial foodborne illness, but also the leading cause of intestinal infection for travelers returning back to the United States. And so there's also a demand for a traveler's vaccine, traveler's diarrhea vaccine.
Now, you mentioned the word glycoconjugate conjugate. And oftentimes when I think of carbohydrates decorating molecules, it's a protein, like a protein on the surface. But sugar molecules can be attached by different organisms to different kinds of molecules?
Correct. And so sugars are typically attached to lipids or to proteins, as you mentioned. And what's been shown is that when we develop antibodies against just a purified sugar, we don't get this long-lasting memory effect and long-lasting immunity to that sugar. But if we conjugate it chemically or biologically, if we covalently attach that sugar to a protein or to a lipid, then you could induce a memory response against that antigen, which gives you longer lasting protection. And so that was one of the key findings early on when people were investigating carbohydrates is how do you induce a long-lasting immune response against these antigens? And the way to do it is to conjugate it to typically a protein.
So when we're talking about the carbohydrates, are those carbohydrates unique to the bacteria, or is the carbohydrate modification being used to make something more antigenic?
It's actually unique to bacteria. So if you look at the evolution of Earth, we know that bacteria have been on Earth for much, much longer than humans. And so the diversity of carbohydrate structures, of sugar structures that are found on bacteria is many, many-fold more complicated than humans. And so there's this opportunity to take advantage of that and target specific bacterial pathogens that cause disease by the unique sugar structures that they have on their surfaces. And so usually when people think of carbohydrates, they think of diet. And we are interested also in how bacteria consume sugars, and we do look at that as well. But when we look at control of bacterial pathogens and how they are able to survive in the environment or cause disease in our bodies, what we're looking at is those unique sugar structures and how they influence the biology of the microbe.
So are there any vaccines that we currently have available that consider the glycobiology of the antigen or how the vaccine is formulated?
Yes, so one of the most common examples that I like to use when I'm teaching is Streptococcus pneumonia, and that's the causative agent of pneumonia. And we currently have glycoconjugate vaccines that are given to infants that encompass these capsular polysaccharide structures that are found on Streptococcus. Now, the complication is that there's over 100 different capsules that coat the different Streptococcus bacteria. And so what industry and scientists have done is they're targeting the specific types that are most dominant in the population. But as soon as you eliminate infection from those, other bacteria have the opportunity to then cause infection. And so we're constantly having to add different sugar structures to that vaccine formulation. And so that's one good example of a glycoconjugate vaccine that's given to infants is against Streptococcus pneumonia.
And the 100 different capsular types, those are differentiated because they have different sugar
Correct.
molecules?
Yes, different sugar molecules as well as decorations on those sugars. But typically it's a different sugar structure that's recognized differently by the immune system.
So thinking about developing a vaccine against, can I say Campy? Do you guys actually call it Campy?
We do all the time, actually, because I always get asked the question of, wow, this microbe is so prevalent worldwide. Why have I never heard of it? And part of the reason, I think, is because it has a complicated name and everyone feels uncomfortable calling it Campylobacter jejuni. So many people, including ourselves, call it Campy. Another reason is because we don't have as many outbreaks associated with Campy. It's really sporadic, and so it doesn't make the news. And so then the general population, I don't think, hears about Campy as much as they would other diarrheal microbes like E. coli or Salmonella.
Are people ever tested for Campy? So like if they go in and they have a diarrheal disease, is that on any of the things that physicians, test for?
Yes, people do test, and it is a reportable disease. And so people are testing for Campy, but typically it's very sporadic. So you go and you eat contaminated chicken somewhere, or the population that actually has the biggest peak of Campy infection are the teenagers that move away from home for the first time and are cooking for their first time. And so those are the people that end up usually with Campylobacter induced diarrhea. But we don't see those big outbreaks that are publicized, although there was one that's mentioned on the CDC website where across North America, pets from pet stores were infected with a multi-drug resistant version of Campylobacter. So Campy, just like other bacteria, is also becoming more and more notorious for being drug resistant. And so there was this drug-resistant isolate that was being circulated, and that was making the news because it was difficult to treat and wasn't self-limiting, as I mentioned earlier on.
So Campy just sounds too friendly. It sounds like a friendly little mascot,
yes,
rather than a deadly pathogen.
Yeah, and and I have a little stuffy in my office of this squiggly little bacteria that sometimes I'll put to my face and make it look like a mustache, just to even make it look cuter. But it does cause serious illness, and like I mentioned, also in some cases paralysis.
So I want to touch on that for a minute, because I remember, so I grew up in Southern California, and I remember my grandparents coming out to visit once, and my grandfather had some illness. I don't remember what it was, but he went on to develop Guillain-Barré, and which I'm sure was really frightening. I don't think it was a severe case, but can you tell us a bit about what's the connection between a friendly thing called Campy and this paralyzing syndrome?
Well, I think it'll come as no surprise to you, since I work on carbohydrates, that there is a sugar link also to this disease. And so many Campylobacter isolates coat themselves with sugars that look like the sugars that we have on our nerves. And so these sugars are referred to as gangliosides. And so many Campylobacters are surrounded with these ganglioside mimics. And so typically when we're infected with Campylobacter, we react against other structures, but not that one. We see that as self. But in a limited number of patients, and it's estimated that it's about 1 in 1,000 patients that are infected with Campy, will see that sugar structure is foreign and will react to it. And when they react to it and form antibodies, those antibodies will then become autoreactive and attack our own nerve fibers. And so you develop this peripheral paralysis known as Guillain-Barré syndrome, which could quickly overtake the whole body, and many of the patients will end up on ventilators. And if it's not treated, it could lead to a significant amount of mortality and definitely morbidity.
So does Campy coat itself with these ganglioside mimics as a way of hiding from the immune system? Is that one of the strategies?
Right. So we think it's beneficial to do so, but because Campy also has been around for much longer than we have, we think it probably serves a different purpose. We don't know why it does it, but it's definitely retained this mechanism of mimicry because it is beneficial. And so we do see that many people that are infected with Campy have these ganglioside mimicking isolates. And we know specifically that those are the antibodies that are causing then the subsequent paralysis, because the treatments for Guillain-Barré syndrome are typically plasmapheresis or IVIG. And so by removing those pathogenic antibodies or competing them out, usually you can get reversal of the symptoms. But in a lot of countries, for instance, we have a clinical study going on right now in Bangladesh that's funded by the Department of Defense. They have a very high rate of Campylobacter infection and Guillain-Barré syndrome development, as does the U.S. military, because they go on maneuvers to several of these low and middle income countries. And so in those cases, the people in Bangladesh, for instance, can't afford plasmapheresis or IVIG. And so they have a lot of lingering effects afterwards. So loss of reflexes, foot drop, or chronic sequelae. So a chronic type of peripheral paralysis that they develop. And so that's why we're very interested in studying it more, to understand why some people see these sugars as self, whereas others see them as foreign, and will go on to develop antibodies against them and attack themselves.
So do you think the increased incidence of Guillain-Barré is directly associated with Campy, or do you think it is due to the fact that Campy may be altering things like the microbiome or the glycome in some way?
We know it's a direct effect of the Campy sugars because they've actually been able to take these sugar structures known as lipooligosaccharides and inject them into an animal and cause paralysis and show that antibodies against those gangliosides are produced. And there's been many, many studies to show that GBS patients have anti-ganglioside antibodies, very high titers of them. And so we know it's directly a consequence of Campy in those cases. We know that there's other microbes that have been associated with Guillain-Barré syndrome as well, but that correlation of why they're causing the disease hasn't been solved yet. So Campy, again, if you go to the CDC website, is the leading cause of GBS. And so that's been firmly established. But there could be other pathogens, other vaccines even, that could be linked depending on how the immune system reacts to those components.
Yeah, I was just thinking about a link with the microbiome because you said that there was a higher incidence, in Bangladesh and then in travelers who may have some disruption of their microbiome that's happening, and maybe that affects how much Campy takes up residence maybe in the microbiome, or how some of the sugars are processed. Maybe it's an additive effect.
Right. No, you're absolutely right, Kim. And that's actually the title of our grant, is looking at how the microbiome could be implicated in Campy disease as well as GBS development. So we are looking at that because one of the findings that we recently came across is that there could be other microbes that also mimic these sugars. And so if you're exposed to these microbes early on in life, maybe that trains your immune system to see it as self, or educates it to react to that sugar if it sees it again. So we're trying to better understand how is it that some people do react to these sugars and some don't, and is there a link to the microbiome? And so this clinical study that we have going on in Bangladesh is doing exactly that, is actually looking at stool specimens from the patients and comparing them to people who've only been infected with Campy and have developed only diarrhea, and versus healthy controls. And we're looking to see, are there any types of population differences? And then looking to see, are those populations then able to recapitulate GBS type disease in a mouse model? And so we're then going to actually do human fecal transplants in mice to see, can we make a mouse more or less susceptible to GBS development?
So Guillain-Barré is basically a autoimmune disease.
Correct.
And I happened to read up on it that it's more common in men, which is kind of against the paradigm of most autoimmune diseases, which are more common in women. Do we have any idea why?
No, there's still a black box as to, why there might be a discrepancy in the sex of the person, and also why certain people react and don't react to this pathogen in the way they do. And so those are still a lot of unanswered questions that gives us a lot of things to work on, and I think is, a very good opportunity for everyone to contribute to the field.
So we've touched a little bit on the microbiome, and people may be familiar with that term, and they also may be more familiar with the genome. But now there's this added layer of the glycome. So could you spend a little bit of time talking to our listeners about what the glycome is and why it's important for human health?
Right. So we think of, as I say, usually sugars from a dietary perspective, but we have a layer of sugars coating every surface. So every parasite you could think of, every microbe you could think of, every cell in our body, that you could picture is covered in a layer of sugars. And so you could imagine if there's so much sugar around that there's going to be a lot of interaction that involves those sugars. So they could be protecting cells or microbes, masking them, as we were talking about in the case of Campylobacter. They play many, many roles in communication between cells, in development, in the case of our bodies. So any disease or healthy state that you could think of is impacted by carbohydrates. And so obviously I have such a strong love for carbohydrates, and so I'm going to tell you how wonderful they are, but they really are something that I think has been neglected when we think about, as you've mentioned, we think so much about nucleic acids, right, and the genome, and we think about the proteome and the proteins, the lipidome and the lipids. We don't think so much about the glycome and how that impacts all aspects of life, but it does. And so it's such an interesting area. And that's why, I love being at the Complex Carbohydrate Research Center, because I'm surrounded by researchers that share the same passion for glycobiology that I do.
I think one issue that makes the glycome harder for people to grasp, even people like me who supposedly has a degree in molecular biology, is that in molecular biology, it's really straightforward. There's a gene, there's a messenger RNA, there's a protein, and you can have alternate splicing and things, but it's very simple. But there's no gene that alone determines a particular carbohydrate structure. And I've been to talks by people at CCRC, and they are tossing out terms of the different sugars and talking about everything that goes in. I don't know if you can quickly give us an update on how a particular structure is, what goes into making a particular carbohydrate structure.
Correct. So, there are multiple enzymes, and it's not intuitive to look at an enzyme and know what it does. So we could predict very accurately, I think, that we've identified a glycosyl transferase, which means a transferase enzyme that could transfer a sugar onto a protein or onto a lipid. But it's much more difficult to know what type of sugar it's actually transferring. And that makes it even more complicated in bacteria, because every time we look at a new microbe, we identify new sugars also that haven't even been described before in nature. And so you really do rely a lot on analytical methods. And so aspects that we use all the time, methods that we use all the time in our lab, are mass spectrometry or NMR. And those help to solve the structures of the sugars and then inform us on what those enzymes could be doing. And so that is why it's complicated, like you say, studying human diseases, for instance, where they have a limited number of sugars that are known, and even more complicated when it comes to looking at bacteria, because they have such an abundant, endless, in my opinion, repertoire of enzymes that could synthesize these different sugar structures. But at the same time, we could exploit them to generate novel therapeutics or vaccines or engineer novel structures. And so I think it's just a gold mine that's out there.
So one of the ways that sugars can be added is through N-linked glycosylation. And you actually discovered this in Campy. It was previously thought that protein glycosylation was really restricted to eukaryotes. So can you tell us how this discovery has changed the technology we use for developing vaccines and therapeutics?
Right. So we know that carbohydrates are very important for function of other proteins. And so in our bodies, a high percentage of proteins are glycosylated. And one of the main functions of those glycans is to protect those proteins from degradation. So many of the protein therapeutics that are available commercially actually require those glycans for those proteins to fold properly, for those proteins to last longer, so have a longer half-life, it's known as. And so we've been trying to recapitulate that by making these glycoproteins in insect cell lines or human cell lines. But by discovering protein glycosylation pathways in bacteria, we can now get bacteria to recombinantly make these glycoproteins for us. And that's become a very inexpensive approach, because now rather than purifying enzymes or synthesizing things chemically, you could actually do a one-pot reaction in a bacterial cell like a simple E. coli and provide it with various enzymes that could then make that therapeutic for you. Of course, we're not at the level of specificity and structure that we need yet. We're still learning, but we can now make N-linked, as you mentioned, Kim, or O-linked glycoproteins in bacteria. And so the difference there is where that sugar is attached to the protein. So in the case of O-linked, typically it's attached to a serine or threonine through a hydroxyl group. And so it's being added to an oxygen. So that's where the O comes from. In the case of N-linked, which is the pathway that we discovered, it's attached to an N, or the amino group of an asparagine residue. And so those are the differences, but there are very different principles involved in how those processes work in eukaryotes. And we're finding more and more complicated methods in bacteria that could be exploited. And so, again, there are such a toolbox for us to use to create novel vaccines, novel therapeutics that we never had available to us before.
I can remember colleagues wanting to recombinantly express a certain protein, and things like yeast were convenient, but yeast glycosylate completely differently, and there would either be no reactivity with particular antibodies, or it didn't work as a vaccine, or it wasn't. If it had a function, it didn't function properly because of this alternate glycosylation.
Exactly. You're absolutely right. And so, there's been companies that have taken advantage of insect cell lines, and insects also make very different structures compared to humans. But they've had to now, over time, engineer those to be more human-like. And so, similarly in bacteria, we could make other bacterial sugars, but it's very hard for us to mimic the types of sugars that are found in humans, because we see a lot more branching, for instance, in human sugars. So the complexity with humans isn't so much the types of sugars that are used, but how they're branched.
So one area of research interest for you is bacteriophage, which, for those who don't know, are viruses that can infect bacteria. So what interests you about these viruses? We're going to switch topics here a little bit.
Yeah, no, that's totally fine. Of course, you know what my answer is going to be: sugars. So, we became interested in it not just from the perspective of phage therapy, which has become much more popular now that we're seeing increased antimicrobial resistance and less antibiotics that we could rely on for treatment, but also from the perspective of why are bacteria making so many different sugars, right? I mentioned to you that humans weren't on this planet originally when bacteria developed these massive repertoires of carbohydrate biosynthesis. What was the purpose of that? And of course, some of it is to resist extreme environments that they might be found in, or to be able to transition from different types of environmental conditions. But we hypothesize that there's probably also a threat of their own viruses, and that they're evolving to be able to avoid their own viruses. And as I mentioned, bacteria, just like eukaryotic cells, are coated by huge layers of sugars. And so these viruses, in order to be able to gain entry into the cell and be able to inject their DNA and take over the host machinery, need to be able to get through that protective sugar layer. So typically, what they're specifically recognizing on that bacterial cell surface are specific carbohydrates. And then they have to have enzymes to possibly cut down those carbohydrates to be able to get down to the surface. And so bacteria have to be able to avoid this and be able to change those carbohydrates. And we've shown, particularly in Campylobacter, but this happens in other bacteria as well, that a lot of the mechanisms that they have for changing their sugar structures are due to pressures on bacteriophage attack. And so if you co-incubate a Campylobacter cell with a bacteria that recognizes its sugar, it has mechanisms to either turn it off or to change it. And we think that's part of the reason this evolution of diversity in carbohydrate structures has occurred, because for every bacteria, there's at least a virus that affects it.
Yeah, and probably multiple.
Correct.
So I have one final question. Do sugars control everything?
Yes. You know what I like to teach is that we're always taught there's 4 macromolecules in life. There's your lipids, there's your amino acids, there's your nucleic acids, and then there's your carbohydrates. So carbohydrates make up one macromolecule class of life that's found in all life forms. But if you then look deeper, we know that most proteins are covered with sugars. They're glycoproteins. We know a lot of lipids are also glycolipids, just like those gangliosides I mentioned on our nerves. And nucleic acids are also made up of ribose, right? So DNA is deoxyribose, and RNA is ribose. And so every macromolecule also involves sugars. So how can you not say it's the most important molecule on Earth?
Okay,
Life really is so sweet.
Yes.
Oh, good one. You have convinced me.
Well, our guest today is Dr. Christine Szymanski from the Department of Microbiology and the Complex Carbohydrate Research Center. Christine, thanks for being with us.
Thank you. It was lots of fun.
Links to additional information about Dr. Szymanski's research can be found on our Instagram account at PPPPodcastUGA.
Thank you for tuning in today. If you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu, and please remember to rate us on your podcast listening software of choice. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and generally keeping us on track.